Okay, hello. I know you guys are thrilled to be learning microbiology from home. And so for the next couple weeks, I will be presenting my material to you guys by voicing over the PowerPoints that you guys already have. So I know I've already covered the first half of Chapter 8. However, to give you guys a little bit of a review so you can go back and listen to it over and over however many times you want, I figured that we would go ahead and just start from the very beginning. So remember, like I said, this chapter is broken down into two parts. The first part of chapter 8 deals with enzymes and why enzymes are important. The second half of chapter 8 deals with glycolysis, the Krebs cycle, and the electron transport chain. So we'll be learning a lot in this particular chapter here. Okay, so remember, there's three definitions total, and remember I said one of these definitions will be on your next exam. So the first definition is metabolism. Metabolism is simply the sum of all chemical reactions in an organism. You guys remember me saying that in a human, it's estimated that there's 10,000 chemical reactions per second. Okay. Also remember I said that the way these processes happen in a human and the way these processes happen in a bacteria cell are pretty much identical. So I'll be referring to these part of the time as they're referred to in humans, and part of the time is how they work in bacteria. All right, so the total sum of all reactions is metabolism. Now, metabolism can be broken down into two types or two parts. There's catabolism and then there's anabolism. The formal definition of catabolism is it provides the release, which is energy and building blocks for anabolism. But remember, I also told you my personal definition of catabolism, and I kind of like my definition a little bit better, but it is the breakdown of large molecules into smaller molecules. So for an example, if you take a protein, and you remember a protein is nothing more than a chain of amino acids. You take that protein and you break it down into these small amino acids. That is an example of catabolism. Now, the opposite of catabolism is anabolism. Anabolism is the building up of molecules. So you start with a whole bunch of small molecules. You put them together to form one large molecule. So again, you can take a whole bunch of small amino acids and you can put them together and they will form a large protein. I always remember this by remembering the phrase anabolic steroids. So bodybuilders would sometimes take these anabolic steroids because they were trying to build up muscle mass. Okay, role of ATP in coupling reactions. First of all, let's talk about ATP. ATP stands for adenosine triphosphate, meaning that there are three phosphate groups. Okay. ATP is energy. This is what gives me the energy to walk. Okay. So the goal of this chapter is to make ATP. Okay. So a metabolic pathway is a sequence of enzymatically catalyzed chemical reactions in a cell. Like I've said, we're going to be studying a lot of pathways in this chapter. We have glycolysis, we have the Krebs cycle, and then we have the electron transport chain. Each one of these is a separate pathway. So a pathway has many steps, and each step of the pathway has its own individual enzyme. So enzymes are going to be a key part of this first half of the chapter, and we'll talk about enzymes more in just a minute. But you can see here in this picture, this is what I was talking about when we were talking about anabolic reactions versus catabolic reactions. At the top, you see simple molecules such as glucose, amino acids, glycerol, and fatty acids. Well, you can take a whole bunch of these glucose molecules and you can combine them together to form a large molecule such as starch. Well, this is the building up. So this is going to be an anabolic reaction. In order for this reaction to take place, you are going to need ATP. One molecule of ATP will be used and is broken down into ADP and PI, which is inorganic phosphate. Now remember, ADP is adenosine diphosphate, meaning that there's only 2 phosphate groups there. You can also, another example of anabolism is with these amino acids. This is the example that I gave on the previous slide. You can take these amino acids and they will undergo anabolism to form a protein. Or you can take one molecule of glycerol and then either 1, 2, or 3 fatty acid chains, combine them together through anabolism, and you wind up with a lipid. So it doesn't always have to be the same type of molecule that's being added together. It can be molecules of different types that are added together. This is still anabolism. Now on the opposite side, we see that these large molecules undergo catabolism, which is the breakdown. And so these large molecules undergo catabolism and they are broken down into their simple structures. So again, you have starch being broken down into glucose. You have your proteins being broken down into your amino acids. And then you have your lipids being broken down into glycerol and fatty acids. So this is all catabolism. And for these reactions to take place, it actually uses ADP and inorganic phosphate, and they come together to form ATP. So these catabolic reactions actually form energy. They make energy, whereas these anabolic reactions actually use energy. Okay, so now let's get to our talk on enzymes and why enzymes are important. But before we start talking about enzymes, let's talk about chemical reactions and rates of these chemical reactions. So the collision theory simply states that chemical reactions can occur when atoms, ions, and molecules collide. So like I used the example in the classroom, I have this box, and inside this box I have all these atoms or these ions or these molecules, and they are moving around. They are not standing still. They are in constant motion. Well, because they are in constant motion, at some point in time, they're going to collide with each other. Okay? That is all the collision theory is stating, that these molecules are in constant motion, and so there is a random interaction that's going to occur where these molecules are going to collide. Well, let's talk about the reaction rate. The reaction rate is simply the frequency at which these collisions are happening. Each collision is a chemical reaction. So each time 2 of these molecules hit each other, that is a chemical reaction. Now, if we wanted to speed up a chemical reaction, how would we go about speeding up a chemical reaction? Well, the classic way of speeding up a chemical reaction is with heat or temperature. Okay? Because if you've ever taken a chemistry class or ever performed a chemistry experiment, a lot of times you'll add heat to the chemical reaction because heat makes the reaction go faster. However, we are talking about biological systems here. We are talking about bacteria cells. We are talking about human cells. Well, we can't heat a bacteria cell up too much, nor can we heat up a human cell, because if we add heat, the cells die. We're not trying to kill the cells here. We are trying to speed up the rates of chemical reactions. So how can we speed up the rate of chemical reactions without adding heat? Well, this is where enzymes come in. So enzymes do the same thing as heat does. They speed up the rates of chemical reactions. Okay. So going back up to activation energy, they have the book definition of activation energy is that it's needed to disrupt the electronic or the electron configurations. However, I also gave you my definition of activation energy, and I kind of like my definition a little bit better. My definition of activation energy is simply the amount of energy needed in order for a chemical reaction to occur. Okay. Okay, so these enzymes are specific. What that means is that enzyme sucrase that we talked about on the previous slide, the only job that sucrase does is it breaks down sucrose. Okay? These enzymes, they are not multitaskers. They only have one job. They do that one job, and that's all they do. So that is what is meant by specific. Okay, next, they are not used up in the reaction. So as you see the chemical reaction and as it's proceeding, that enzyme is not part of the chemical reaction. And this is a very good thing because that allows these enzymes to be reused over and over again. So they're able to be recycled. One enzyme molecule could potentially be used 100 times or even 1,000 times before it's just kind of naturally degrades. Now, the benefit of this, think about it. If your body had to make a new enzyme every time an enzyme was needed, your body could not keep up with the demands of the enzyme. And so by being able to reuse these enzymes, our body as well as bacteria cells are able to fully function. Okay, 90% of enzymes are proteins. This, like I said, is a test question on your next exam. Okay, so again, remember though, a protein is nothing more than a chain of amino acids. Okay, so that becomes important in a few minutes. We're going to talk about something else. But for the most part, these enzymes are proteins. Okay, let's look at the enzyme components or the different parts that make up an enzyme. Well, you can look at the picture in the upper right hand corner. I also said that this picture would be on your next exam as well. Well, the biggest part of the enzyme is known as the apoenzyme. The apoenzyme, first of all, it's the largest part. It is composed of protein, but look, it is inactive. By itself, it is not functional. It has no function. That apoenzyme has to be activated before it's functional. So it's usually activated by one of two different things. It's either activated by a coenzyme or a cofactor. One or the other, never both. These are both the active portions. So how do you know if something is a coenzyme or how do you know if something is a cofactor? Well, if you look at the list of cofactors on the PowerPoint, iron, zinc, magnesium, calcium, these are all straight off the periodic table. These are all elements straight off of the periodic table. However, if you look at the list of coenzymes, we have NAD+, NADP+, and FAD. Remember, we talked a little bit about these already. NAD+, yes, these are acronyms. NAD+ stands for nicotinamide adenine dinucleotide. For that reason, because they have really long names, you don't have to learn the really long names. You will see NAD+ again when we talk about the Krebs cycle and a little bit in glycolysis. You will see FAD again when we talk about the Krebs cycle. You will not see NADP+ again, simply because you only usually see that one in photosynthesis, and we don't talk about photosynthesis in this class, so you won't see NADP+ again. But anyway, NAD+ and this FAD, these are known as coenzymes. So you're either going to have a cofactor or a coenzyme. And it's going to bind to the apoenzyme to form the holoenzyme. Holoenzyme, well, it sounds like whole enzyme. It is the apoenzyme plus either a coenzyme or a cofactor. So this is a whole enzyme. It is active. It is now ready to work. Each enzyme has what's known as an active binding site. In this holoenzyme, it's that groove at the top. you see the substrate in red, it will bind to that active binding site or to that groove. Now, the substrate, I'll give you an example. When we were talking about sucrose binding to the enzyme sucrase, well, if that were the case, our substrate would be sucrose, and our enzyme would be sucrase. Naming of enzymes. Like I said, usually enzymes end in ASC, as you see down here in red. And the name of the enzyme usually kind of tells you what it does. So lactate dehydrogenase. Well, it sounds like dehydrogen, and that's exactly what it does. It removes a hydrogen from lactate. Also, remember that they usually end in ASC because that's also going to be a test question. I'm going to give you a question and I'm going to say which of the following is not an enzyme. So all you have to do is you look at your list of words, find the one that does not end in ASE, and that's going to be your answer. Now, it is estimated that there's 100,000 different enzymes that exist. But all of these enzymes can be broken down into 6 main categories or classes. That is also a test question. There are 6 main classes of enzymes. All right, now let's look at this slide. This slide is nothing but review. There's nothing new on this slide. It's just going through a nice little review for you. So here you have your enzyme. Here you have the active binding site of the enzyme. And here you have the substrate. So they're using the substrate sucrose so that we know that this enzyme would have to be sucrase. Well, the substrate, it binds to the active binding site of the enzyme, forming what's known as the enzyme substrate complex. The enzyme, it does its job, it does whatever it's supposed to do. In this case, it breaks that complex sugar into 2 simple sugars. And then it releases the products. So you have 1 molecule of glucose, 1 molecule of fructose, and this molecule of enzyme will go back. And like I said, they are recycled. This molecule of sucrase will now pick up the next incoming molecule of sucrose, and the entire pathway starts again. Okay, sorry about that. So like I was saying, these enzymes are nothing more than proteins. And these proteins, they are nothing more than chains of amino acids. Now, a typical protein, the active functional protein, you see it has some alpha helixes in there, and you see it has a miniature beta pleated sheet. But this is the entire protein, and remember, you have all these bonds that are holding a protein together. You have covalent bonds, you have ionic bonds, you have hydrogen bonds, you have van der Waals forces, you have disulfide bridges. So you have all these bonds that are coming together to hold that protein in its unique shape. Well, a common factor that influences enzyme activity is temperature, mainly heat. When you add heat to an active functional protein, that heat breaks all the bonds that's holding that protein together. That protein will unravel to the denatured form of the protein. Okay. Now, this protein is still the correct amino acid sequence, but it's not in its normal 3-dimensional shape. Therefore, it is a non-functional protein. And because these enzymes are proteins, adding heat to these enzymes inactivates the enzymes. So this is one reason why we don't want fevers to get too high. Another thing, we don't want bacteria cells to get too hot unless our goal is to kill them. If we're trying to kill them, heat is great. But if we are not trying to kill them, we do not want to add too much heat to their system because heat starts breaking bonds. Now, in addition to heat, pH also has the same effect, particularly acids. Acids work by breaking the bonds that hold the functional protein together, and these acids break those bonds, causing the active protein to unravel into the denatured form of the protein. Either way, it's still inactive. It does not work anymore. Okay, let's look at the substrate concentration in the bottom right corner. I also said on your next exam, there is going to be a picture very similar to this one, and I'm going to ask you what's going on in this picture. All right, so this picture is talking about substrate concentration and how the enzyme is maxed out. It's basically asking why this red line, why the red line plateaus, why does it flatten out? Okay, so the x-axis is substrate concentration, the y-axis is enzyme concentration. Well, because we've been talking about sucrose so much, let's just continue with our talk on sucrose. Let's say that we have 1,000,000 molecules of sucrose. And as far as the enzyme is concerned, the y-axis, what if we only have 10 molecules of the enzyme? So we have 1,000,000 molecules of sucrose. We only have 10 molecules of the enzyme sucrase. Well, what this picture or what this graph is showing is it really doesn't matter how much substrate you have. What matters is how much enzyme you have. You only have 10 molecules of this enzyme, sucrase. So that means the chemical reaction will only occur until all 10 of those active binding sites on the enzyme molecules are full. Once all of those 10 molecules of enzyme, once they are taken up, there's no more enzyme to do work. And so the red line plateaus. That is what is meant by the enzyme being maxed out. So again, it's almost like a rate limiting step in chemistry. The reaction is only going to progress as much as the least concentrated component. And in this case, the least concentrated is the enzyme. So once all the molecules of those enzymes are full, it doesn't matter how much substrate you have. When all the enzymes are full, no more chemical reactions can occur, and so the line plateaus. Okay, so let's move on and talk about enzyme inhibition. Enzymes are great and all, and normally we want enzymes to function. However, there are a couple of ways in which we can stop enzyme activity. Sometimes this is good, sometimes this is bad. So let's talk about normal activity, and then let's talk about how we can stop these enzymes from working. All right, so the first picture on the far left, this shows what is normally supposed to happen. We have an enzyme, we have an active binding site, and it shows the substrate binding to the active binding site on the enzyme. Okay, again, this is what is normally supposed to happen. Well, the first type of inhibition that we're going to talk about, and again, inhibition just means we are stopping that enzyme from working. So the first type of inhibition that we are going to talk about is competitive inhibition. So here it shows the enzyme, here it shows the active binding site, but look, there is now a yellow molecule. And that yellow molecule, it could be a lot of different things. Examples, if these are poison, such as cyanide, arsenic, mercury, they would work by binding to the active binding site of the enzyme, so that now the normal substrate, when it comes to bind, it cannot bind there anymore because that yellow molecule is there. This is called competitive inhibition because both the substrate and that yellow molecule are both competing for the same binding site. Now you see this arrow going back out, meaning that sometimes the substrate can't bind, so it goes back out into the environment. Sometimes this is reversible, If it's reversible, what that means is that at some point in time that yellow molecule will then leave, freeing up the active binding site, so then the substrate can then come in and bind. However, if this is non-reversible, what that means is that that yellow molecule is there for good. And because it's there for good, that enzyme will no longer function anymore ever again. So basically this enzyme will be useless. Now, the other type of inhibition is known as non-competitive allosteric inhibition, and it's seen over here on the far right. Also, I should have mentioned this at the beginning. This picture here, this entire picture, will also be on your next exam. Okay, some enzymes, not all enzymes, have what's known as an allosteric site. An allosteric site, sometimes it's called a secondary site, but it's an alternative binding site. Okay, you can see that little groove down at the bottom of the enzyme. That is the allosteric site. Okay, so when allosteric inhibition occurs, well, there's a yellow molecule that binds to the allosteric site, and then look what happens. When the allosteric binding site is full, the active site changes shape. It goes from this to this. Okay? And now the substrate, when it comes in to bind, it won't bind because it doesn't fit anymore. And so the arrow, it shows the substrate being sent back off into the environment because it simply doesn't fit. Now, at some point, this yellow molecule, it may leave, meaning it's reversible. If it leaves, then the enzyme will go back to the normal shape. The active binding site turns back normal, and then the substrate would be able to bind there once again. But as long as that allosteric site is full, no binding can occur. So it is called non-competitive inhibition because the substrate and that yellow molecule are not competing for the same binding site. Okay, so these are two mechanisms that are used to inhibit or to stop enzyme activity. We have competitive inhibition versus non-competitive allosteric inhibition. Okay, now let's move on and talk about feedback inhibition. Notice on the left, we have a pathway. I said pathways are usually multiple steps, and each step of the pathway usually has its own enzyme. Well, here you see enzyme number 1. It has an active binding site. The substrate comes in and binds to the active binding site. Enzyme number 1, it does its job. It does whatever it's supposed to do, and then it releases the substrate. That substrate is now known as intermediate A. So there's enzyme number 2 that comes in. It takes intermediate A. Okay. Enzyme 2 does its job. It does whatever it's supposed to do. It changes intermediate A and then it releases it. It is now known as intermediate B. And then we have a third enzyme that comes in, grabs hold of intermediate B. It does its job, does whatever it's supposed to do, and releases the product. Well, the end product looks like this. It started way up here at the top. The substrate looked like this. But the substrate, with the help of those 3 different enzymes, each enzyme changed the substrate somehow, so that at the very end of the pathway, you're left with the end product. Now, remember, we're talking about bacteria cells here. Bacteria cells, for the most part, are free empty space on the inside. They are not compartmentalized. So what if this end product is continuously being made, but that bacteria isn't using the end product at that moment? Well, what's going to happen is that end product is going to continuously be made. It's going to waste energy. It's going to waste resources because it's being made, but it's not being used. It's not being needed at the moment. So it's going to, the concentration of that end product is going to continuously and going to continue to build up inside the bacteria cell. Well, notice the end product, because it's free floating, it will eventually make its way back to enzyme 1. It just so happens that enzyme 1 has an allosteric binding site. The end product goes back and it binds to the allosteric site of enzyme 1. When binding happens at the allosteric site, the active site of the enzyme 1 changes shape. So now the normal substrate can no longer bind to enzyme number 1 because it doesn't fit. Because it doesn't fit, enzyme 1 no longer operates. So enzyme number 1 has been shut down. Well, if enzyme 1 is shut down, that also means that enzyme 2 and 3 are going to be shut down as well. The entire pathway has stopped because the substrate can no longer bind to enzyme number 1. Okay, now later on, as the bacteria cell starts using the end product, the concentration of that end product will decrease. It'll be used up and it'll get low enough. This is a reversible binding here. So the end product will be released from the allosteric site. When that happens, the active site of enzyme 1 will change shape back to its original shape. Once it's back in its original shape, the normal substrate can then once again bind to enzyme number 1, and the pathway will start up again. So this slide talks about allosteric inhibition and how you can shut down a pathway when it's not needed through allosteric inhibition. Okay, so our talk on enzymes is done. There are 2 topics that we need to talk about that will get you ready for glycolysis and the Krebs cycle and the electron transport chain. The first of those 2 topics is the idea of oxidation reduction. So oxidation reduction reactions usually occur together. They usually are simultaneous reactions, meaning oxidation occurs and then reduction immediately follows. So they took those 2 terms, oxidation and reduction, and they pushed them together to form one word, and that one word is known as a redox reaction. So redox simply means it's oxidation and reduction happening at the same time. Now, oxidation reduction, this is a chemistry, this is something that's typically taught in chemistry. Oxidation is the removal or the loss of electrons. Reduction is the gain of these electrons. It seems fairly simple and fairly straightforward, but if you look at it, if you look at the term reduction, well, it sounds like reduce. Reduce makes it sound like lowering or losing something, but it's actually the gaining of electrons. So it's the opposite of what you would normally think. And so because of that, there are 2 mnemonics that are used, and remember I wrote these on the board for you guys. The first one being oil rig, oil, O-I-L. Oxidation is loss. Rig, R-I-G. Reduction is gain. And then I told you about the other mnemonic, the one that I use, talks about Leo the lion goes grr. Leo, loss of electrons is oxidation. Grr, which is G-E-R, gain of electrons is reduction. I don't care which one you learn, but I highly recommend that you learn one of them, because oxidation reduction seems easy when you have the information right in front of you, but on an exam or when you're looking at pictures, it's not quite as easy. So if we look at this oxidation reduction reaction that's going on here, they did it in a very simple format. they're just using colored balls or colored spheres. So over here in the reactant, so this is showing a chemical reaction that's occurring, and you have your reactants on the left-hand side of the arrow, and then you have your products over here on the right side of the arrow. Well, in the reactants, you see this yellow molecule, and it's showing an electron here. Well, then if you follow it over to the product, it lost that electron. Loss of electrons is oxidation. So that product, that yellow molecule, has been oxidized. Now, if you look at that bluish purplish sphere labeled B, well, in the reactants, it's by itself. But if you follow it over to its products, you see that it has gained an electron. Gaining of electrons is reduction. So in the product, that purplish blue sphere has been reduced. Now, let's move on and talk about oxidation reduction reactions in a biological sense. First and foremost, note up here, in biological systems, the electrons are often associated with hydrogen atoms. So go ahead, and I told you to write in your notes, 1 hydrogen equals 1 electron. And the reason you're able to do that is if you look at the periodic table in chemistry, if you find hydrogen, well, hydrogen is the simplest element. Hydrogen has 1 proton, 1 electron, and 0 neutrons. So 1 hydrogen equals 1 electron. So you are going to see with oxidation reduction reactions in biological systems, you are going to be seeing hydrogens move. But you have to remember that each hydrogen is an electron. Okay. So if we look down here at the bottom at this chemical reaction that's occurring, it's showing you a yellow molecule, and all it says is it's some type of organic molecule. Well, remember, the definition of organic just means that it is something that contains carbon. We've been talking about sugar pretty much this entire chapter, so let's just go with sugar. Let's pretend that molecule, that organic molecule, is glucose. Well, the chemical formula for glucose is C6H12O6. So this is glucose, and it's showing 2 hydrogens. Remember, there's a total of 12 hydrogens on glucose. This is just showing 2 of those 12 hydrogens. Well, if you look at it, follow the yellow molecule over to the product side, it lost those 2 electrons. Loss of electrons is oxidation. So in the product, that yellow molecule has been oxidized. Okay. All right, now let's look at the oval. It is a bluish purple oval. It says it is NAD+, which is a coenzyme. We just talked about NAD+ a few minutes ago. I said you would see it again. Okay. Not only is it a coenzyme, but underneath it in parentheses, it says that it is an electron carrier. It carries electrons in the form of hydrogen. So its job is to pick up hydrogens. Okay. All right. Well, if you notice it on the left-hand side of the arrow in the reactant section, you have NAD+ by itself. If you follow it over to the product, it gained 2 electrons. Okay. It gained 1 full hydrogen, and the hydrogen shows its electron. But then it just stole the electron of that other hydrogen because the other hydrogen is out here, and it's showing H+. The only way you can have a hydrogen with a positive charge is if it lost its electron. And it did lose its electron. Its electron is right here. So the hydrogen, so this NADH was reduced because it gained an electron. NADH was reduced, NAD+ was reduced to NADH. Okay? I told you it picks up hydrogen. It picks up electrons in the form of hydrogen. Okay? So that NAD+ was reduced to NADH. So this is oxidation reduction reactions in biological systems. You will see hydrogens moving. You have to keep in mind every hydrogen has an electron. So when those hydrogens are moving, those electrons are also moving with it. Okay, the next topic is the idea of phosphorylation. Phosphorylation simply means you're adding on a phosphate group to something. Okay, but now there's three types of phosphorylation that I'm going to talk about. The first type is substrate level phosphorylation. We'll talk about that in just a minute because you'll see a picture. The picture down here at the bottom is talking about substrate level phosphorylation. We will also talk about oxidative phosphorylation, but that won't occur until we get to the electron transport chain. Therefore, I'm not going to talk about it right now. The third type of phosphorylation is known as photophosphorylation. You see, I don't give you any information about it because this only occurs during photosynthesis, and I don't really cover photosynthesis in this class. So for the exam, make sure you know that there are three types of phosphorylation, but you only have to know details really about the first two types of phosphorylation. So let's get started by talking about the first type of phosphorylation, which is known as substrate level phosphorylation. The definition is transfer of a high energy phosphorus to ADP. So basically what that's saying is anytime you have ADP, which is adenosine diphosphate, and you're adding phosphate to make ATP, that is going to be an example of substrate level phosphorylation. So before I show you the picture which talks about substrate level phosphorylation, let's go up here and talk about ATP. This picture at the top, this is showing ATP. You have an adenosine group followed by 3 phosphate groups. Now the only interesting thing about this picture here are some of the bonds. Now a normal bond in chemistry is represented by a straight line. However, you notice that there are 2 wavy bonds in here. Those are a special type of bonds. Those are high energy bonds. What that means is if that bond is broken, an excessive amount of energy is released. So that is what is special about those particularly wavy lines. Those are high energy bonds. All right, now let's look at a particular example of substrate level phosphorylation. This can be seen here in the picture in the bottom. Remember, the definition is ADP plus P equals ATP. That is substrate level phosphorylation, and that is exactly what is occurring here. You have ADP, adenosine diphosphate, di because you have 2 phosphate groups, plus incoming phosphate equals adenosine triphosphate. Now you have 3 phosphates. So ADP plus this equals this. This is the example of substrate level phosphorylation. ADP plus P equals ATP. Well, if you look at the definition of substrate level phosphorylation, that's the exact definition. ADP plus P equaling ATP. Make sure you remember that, because when we get to glycolysis and the Krebs cycle, we will see examples, specific examples of substrate level phosphorylation. And you will have to make sure you understand what substrate level phosphorylation is so you can point out to me when it's happening. Okay, so we are about to start talking about glycolysis. But before we talk about glycolysis, let me just give you a little bit of background information on glycolysis. Remember, glycolysis is the breakdown. We're going to start off with the sugar glucose, and it's going to be broken down into pyruvate. So this is an example of catabolism, because remember, catabolism is where you start off with a larger molecule and it's broken down into something smaller. Okay, we are going to be talking about 3 types of metabolic pathways. We are going to be talking about aerobic respiration. We'll be talking about anaerobic respiration, and then we will be talking about fermentation. Probably 90% of my talk is going to be on aerobic respiration, because if you understand aerobic respiration, you will automatically understand anaerobic respiration and fermentation. Aerobic respiration. So most bacteria need oxygen, just like we need oxygen. This is where aerobic respiration comes in. It means oxygen is needed. Anaerobic respiration. We haven't really talked about them yet, but we will in a later chapter. There are bacteria that die in the presence of oxygen. Oxygen is toxic to those bacteria. They're going to use anaerobic respiration. Remember, the prefix a in front of a word means without. So this literally translates into without oxygen. Okay. And then the third type is fermentation. We are very thankful for fermentation because a lot of bacteria are able to ferment and make certain end products, and these end products are commonly used in the food industry. So again, we'll be talking more about fermentation. So as far as aerobic respiration is concerned, it's broken down into three main parts. And there's a total of 26 to 28 steps involved in these three main parts. The first main part is glycolysis. Glycolysis is defined as the oxidation of glucose to pyruvate. Okay. So note, it is the oxidation of glucose and only glucose. There's about 30 different types of sugar. We have glucose and fructose and lactose and mannose and dextrose and maltose, and the list continues. However, only glucose can enter into glycolysis. Okay. So it is called the oxidation of glucose. Remember, oxidation is loss of electrons, because we are starting off with glucose, and glucose is broken down into pyruvate. During this process of glucose being broken down, hydrogens are lost. Well, those hydrogens carry electrons, so you're losing electrons each step of the way. So this is a definition of oxidation. The end product of glycolysis is pyruvate. Okay. And you will see that in just a few minutes when I show you the slide of glycolysis. Now in the upper right hand corner you see glucose. Glucose naturally exists in two forms. About 50% of the time glucose exists as a straight chain form. The other 50% of the time glucose exists in a ring form. And these two are at chemical equilibrium with each other, meaning in nature they can easily convert back and forth between one form and the other. And this is important because only the straight chain form of glucose can enter into glycolysis. Okay, so step 2 is the Krebs cycle. The Krebs cycle takes the product of glycolysis, which is pyruvate, and it converts pyruvate into acetyl-CoA. The acetyl group enters into the Krebs cycle, and this is where the majority of energy is made. The Krebs cycle makes ATP. This is energy in its final form. It is ready to be used. But the Krebs cycle also makes NADH and FADH2. This is what I consider to be intermediate energy. In the electron transport chain, it will be converted to ATP, but right now it stays in the form of NADH and FADH2. That's why I call it intermediate energy. The third main phase is where oxidative phosphorylation occurs in the electron transport chain. This is where all of those molecules of NADH and FADH2 are made, and they are shipped off to the electron transport chain where then they are converted into ATP. And again, total from start to finish, it's about a 26 to 28 step process that starts off with glucose and you wind up with energy. So the end product is energy. And remember, energy in its final form is ATP. But NADH and FADH2 will also be converted into ATP later on in the electron transport chain. Okay, so let's start talking about glycolysis first. I've already said that glycolysis is a multi-step breakdown of glucose into pyruvate. During glycolysis, you make a small amount of ATP, usually 4, and you make a small amount of reducing power. Reducing power is NADH. Okay? So you make a small amount of NADH during glycolysis. Now, alternate pathways to glycolysis. Well, one alternate pathway to glycolysis is known as the pentose phosphate pathway. And this pathway is very useful because, like I said, only glucose can enter into glycolysis. A lot of sugars are transformed into glucose so that they can enter into glycolysis. However, there are 5 carbon sugars. 5 carbon, the prefix pent means 5. There are 5 carbon sugars such as fructose, fruit sugar, which cannot undergo glycolysis. So they're still able to make energy, but instead of using glycolysis, they're going to use the pentose phosphate pathway. So it's a pathway designed for the 5 carbon sugars. Another pathway is known as the Ettner-Dwaderoff pathway. The only thing you have to know about this is it is an alternative pathway that some bacteria use instead of using glycolysis. For example, E. coli. E. coli they do not undergo glycolysis. E. coli uses the Ettner-Dwaderoff pathway instead. And that's all you have to know about the 2 alternative pathways. All right, let's get into the steps of glycolysis. And I know I covered this in lecture, but luckily I have the opportunity to go over it again, simply because it's a lot of information. There's a lot of things happening. And so let's start at the very top. Remember, only glucose can enter into glycolysis. Well, over here on the far right up here, it's showing the full structure of glucose. However, when the manufacturers or the publishers of this book, they attempted to make this slide a little easier by just showing you the 6 carbon backbone. So if you go through glucose and you count the carbons, well, there's 1, 2, 3, 4, 5, 6. There's those 6 carbons right there. This is the carbon backbone. They are not showing you the oxygens or the hydrogens that are also there. And again, they did this to simplify the picture, but there's a couple of steps in here where it's kind of confused students. And again, I'll talk about where those steps occur. So glucose enters into glycolysis, and the first thing that happens is in step 1, glucose is converted into glucose 6-phosphate. Well, look, glucose 6, on that 6th carbon, a phosphate was added. That's the only thing that's happened. A phosphate was added. Well, you have to ask yourself, where did the phosphate come from? Look on this over on the left side. Over here, you see adenosine triphosphate was converted into adenosine diphosphate. ATP lost a phosphate group. That is that phosphate group right there. That is where that phosphate group came from. ATP lost it. Okay. The next step, glucose 6-phosphate is converted into fructose 6-phosphate. This is one of the steps that kind of confuses students because it looks like they have the same molecular structure, but they have different names. The only thing that happened here is molecular rearrangement. Remember, it's just showing you the carbon backbone. It is not showing you all of the oxygens and all of the hydrogens that are also attached. If you were to Google those two structures, you would see that they actually have a different structure. Okay, going to the next step. Fructose 6-phosphate is converted into fructose 1,6-biphosphate. And let me see here. I think I might be able to add like a pointer or a highlighter to kind of keep up with the steps that I'm on. Okay, so I'm right here now. So fructose 1,6-phosphate, or fructose 6-phosphate was converted into fructose 1,6-biphosphate. Biphosphate means two phosphate groups. Well, now there's a phosphate group on the first carbon, and there's a phosphate group on this last carbon over here. Okay. Well, you have to ask yourself, where did that phosphate group come from? It came the same place that it came up here. A molecule of ATP was put into the system. That terminal phosphate group was cut or cleaved. ATP was converted into ADP. The phosphate group that ATP lost is now right here. Okay? So now we are here. We are at fructose 1,6-biphosphate. So if you look at the structure, it's a 6-carbon backbone, and it's got a phosphate at both ends. This molecule gets cut in half. Right through here, it gets cut in half. Okay. The left side of the molecule becomes glyceraldehyde 3-phosphate. The right side of the molecule becomes dihydroxyacetone phosphate. Okay. Well, remember, like I said in class, luckily for us, these two molecules of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, they are at chemical equilibrium with each other. And what that means is glyceraldehyde 3-phosphate can easily convert and become dihydroxyacetone phosphate. And dihydroxyacetone phosphate can easily convert and become glyceraldehyde 3-phosphate. Okay? For that reason, to make this picture simpler, let's pretend that when this molecule of fructose 1,6-biphosphate up here, when it got cut in half, let's pretend that both halves of that molecule followed this pathway and became glyceraldehyde 3-phosphate. Okay. All right, so now we have two molecules of glyceraldehyde 3-phosphate because this molecule up here cut in half, they both followed the left pathway. So now we are at a 3-carbon structure with one phosphate group here on the end. Well, look at the next step. Okay, I'm not going to lie, there's a couple different things that's going on here in this step. You have a 3-carbon backbone with a phosphate. Well, look down here. You have a 3-carbon backbone, still has the phosphate from the previous step, but now it has a new phosphate. It has this phosphate right here. All right, well, where did the phosphate group come from? Well, remember, there are free-floating phosphates because this is bacteria. There are free-floating molecules on the inside of the bacteria. There are no compartments. Well, the first step, this carbon right here, I'm going to change my highlighting colors. This carbon right here, where I'm putting the purple line above, that carbon right there lost hydrogen. Okay. It lost hydrogen, and now there's a space for the phosphate to bind. So that terminal carbon here lost hydrogen, which gave that phosphate a spot so it could bind. And that is this phosphate right here. Okay. All right. So just to kind of erase everything and keep everything clear here. All right. So after that carbon lost the hydrogen, you have to ask, and now there's a space for the phosphorus to bind. It bound to the carbon that lost the hydrogen. Well, you have to ask yourself, okay, that carbon lost hydrogen, but where did the hydrogen go? Well, here you see. Remember, I said you would see it again. NAD+. It is a coenzyme, but it is also an electron carrier. It picks up hydrogen in the form, or it picks up electrons in the form of hydrogens. Okay. So NAD+ comes in, takes those hydrogens that this carbon right here lost, and the NAD+ is converted to NADH, and this molecule of NADH is then shipped off to the electron transport chain. So again, this molecule of NADH is shipped off to the electron transport chain. Okay, we are here now. Okay, we have a 3-carbon backbone with a phosphate at both ends. Well, going through step 6, look down here. We have a 3-carbon backbone with 1 phosphate group. All right, well, it lost a phosphate. So you have to ask yourself, well, it lost a phosphate, where did it go? Well, you have a molecule of ADP, adenosine diphosphate. It comes in, it takes this phosphate group, and ADP is converted into ATP. Remember when we were talking about substrate level phosphorylation, I asked you, don't forget what substrate level phosphorylation is. Anytime you have ADP taking a phosphate and becoming ATP, that is substrate level phosphorylation. Well, that's exactly what you have here. ADP took this phosphate and was converted into ATP. That is substrate level phosphorylation. This is the first time that we've actually seen substrate level phosphorylation occurring. Okay, we are here now. We have a 3-carbon backbone with a phosphate group. Well, look what happens. The phosphate group, the only thing that happened is molecular rearrangement. The phosphate group here on the end, it got moved down here to the center. Water was lost as a byproduct, but nothing really special happens, just molecular rearrangement. And now we are here. We have a 3-carbon structure with phosphate in the middle. Well, step 9 shows you start here and you go down here. Well, now you have the 3-carbon backbone and it lost its phosphorus, or that phosphate group. So like before, we have to ask ourselves, where did the phosphate group go? Over here on the side, you see once again ADP coming in, stealing the phosphate group, which converted then into ATP. This is another example of substrate level phosphorylation. So we have seen substrate level phosphorylation occurring twice now. Here at step 6 and here again at step 9. And now we are at the very end of glycolysis, which is with the product pyruvate. Now, remember, pyruvate, the full structure of pyruvate can be seen here. It's got carbons, it's got hydrogens, it's got oxygens. However, you see here, only that carbon backbone is being shown. Okay? Again, they're doing this to try to keep things simple. Also, remember from your notes, if you didn't write this down, make sure to write it down. This slide, they're calling the end product pyruvate. But on the next slide, they're calling it pyruvic acid. Remember, pyruvate and pyruvic acid are the same things. Okay, The only other thing that I want to talk about before I move on to the next slide, let's talk about what all is made. Well, here you made one molecule of NADH. Remember, we called this reducing power. So we made one molecule of NADH. We made one molecule of ATP here. And then down here, we made another molecule of ATP. So we made NADH, ATP, and ATP. But remember, this molecule up here at the top, fructose 1,6-biphosphate, remember, it got cut in half. So anything that we made on the left-hand side, we have to multiply by 2. So we made one molecule of NADH, but don't forget, multiply it by 2. 1 times 2 is 2. So we actually made 2 molecules of NADH. And then when we look at ATP, well, we made 1, 2, but we have to multiply that by 2. So 2 times 2 is 4. We actually made 4 molecules of ATP. Okay, so we made a total of 4 molecules of ATP. But what if I ask you the net amount of ATP made? The key word being net. Going back up here to the very top, right here and right here, we had to use 2 molecules of ATP to even get glycolysis started. So although we made 4 molecules of ATP, we had to use 2. So 4 minus 2 is 2. So on the exam, if I were to ask you how much net ATP is made during glycolysis, the answer is 2. Okay? Okay. Okay, hello again. I'm sorry for the delay, but I will make sure to finish up chapter 8, and then we will start on chapter 9 next. So we covered this slide in class as well. Hopefully you've had a chance to review my previous lesson and you understand the glycolysis a little bit better. Remember that this is the last step of glycolysis. So this slide right here is showing you, I call it the transition step. It's preparing the product of glycolysis, getting ready for it to enter into the Krebs cycle. So if you notice, I said you have 3 different things going on. Remember, the end product of glycolysis is pyruvate. And I said that pyruvate and pyruvic acid are the same thing. So on this slide, they're calling it pyruvic acid. Well, if you look at the beginning, it's pyruvic acid, and then if you see where it goes, it forms into something known as acetyl-CoA. Well, okay, so if you look at the pyruvic acid, it has 3 carbon backbones. Each one of those balls represents a carbon. And then after it goes through that transition step at the very end, it has a 2 carbon structure attached to a CoA. So the first thing that happened is a carbon was lost as pyruvic acid was going through that transition step to become acetyl-CoA. This carbon over here was lost in the form of carbon dioxide gas. Okay, the next thing that happens, you see here CoA. Remember, CoA is a coenzyme. It comes in at this step and it binds because this carbon is now lost. So the CoA comes in and it binds to that 2 carbon structure. And that is what you see down here. You see acetyl-CoA. Okay, now if we look on the left side of that arrow, you see NAD being converted to NADH. Well, remember, when carbon is lost, hydrogen is also lost. That NAD+, it comes in and it steals that hydrogen, and the NAD+ is converted to NADH. Remember, NADH, this is energy in its intermediate form. But first, before it's transferred or translated into actual energy, this molecule of NADH will have to be shipped off to the electron transport chain. Okay, notice we have acetyl-CoA as the end product, but then if you follow this arrow here, it shows this arrow of CoA leaving. So that CoA is only good for one step. It comes in up here, it does what it needs to do, and then it leaves down here. Excuse me. So that molecule of CoA is recycled back up top, and it's able to be recycled and reused over and over again. Because it is only this 2-carbon structure, which is known as an acetyl group. This is a chemistry term. This 2-carbon structure, which is known as the acetyl group, this is the structure that enters into the Krebs cycle. Now, other names for the Krebs cycle, if we go up here, other names for the Krebs cycle include the citric acid cycle. And it's called the citric acid cycle because the first product of the Krebs cycle is citric acid. Okay. It's also known as the TCA cycle. TCA because it stands for tricarboxylic acid cycle. So those are the 2 other names. Make sure you know the 2 other names for the exam. All right, let's go ahead and move on and talk about the next slide, which is the Krebs cycle. Okay, so this is the Krebs cycle, and remember, this is a simplified version of the Krebs cycle. It's just showing you the carbon backbone. It's not showing you those oxygens or those hydrogens. It's also not showing you the names of the intermediates. It's not showing you the names of the enzymes. So it's basically just wanting you to follow that carbon backbone here. So this top step right up here that I'm circling, this is the step from the previous slide that we talked about. We have pyruvic acid, which is the end product of glycolysis. And I said there's 3 different things happening. The first thing is that you lose a molecule of carbon as carbon dioxide. The acetyl-CoA, or the CoA, comes in and it binds to the remaining acetyl group, and then the NADH, it steals hydrogen. As the carbon is lost with CO2, it steals hydrogen, and the NAD plus is converted to NADH, and this molecule of NADH is then shipped off to the electron transport chain. So we covered everything that's in the circle here. We covered this on the previous slide. Okay, so now let's talk about what's going on with the Krebs cycle. So I said it is this acetyl group, literally this 2-carbon structure right here, that enters into the Krebs cycle. Okay. It enters into the Krebs cycle, and it binds with the last product of the Krebs cycle to form the first product of the Krebs cycle. So basically, this plus this equals this. Okay. So your acetyl group plus OAA equals citric acid. In case you're wondering, OAA, they did give you the abbreviation for the name. This stands for oxyl acetyl acetate. You do not need to know that for the exam, but they just put that information in here, so in case you were wondering what OAA stood for, well, that's what it stands for. Okay, so like I said, the first product of the Krebs cycle is citric acid. All right, and I said that's also a test question. The first product of the Krebs cycle is citric acid. Notice that citric acid has a 6-carbon backbone structure. Well, the 6-carbon goes to a 5-carbon structure, meaning it lost a carbon. What happened to that carbon? Well, that carbon was also lost as CO2. As carbon is lost, hydrogen is also lost. Therefore, NAD plus comes in, steals the hydrogen that was lost, was converted to NADH, and then this molecule of NADH is then shipped off to the electron transport chain. Okay, so we are here. We have a 5-carbon structure. Well, the 5-carbon structure is going to a 4-carbon structure. So again, a carbon was lost in the form of CO2. As carbon was lost, hydrogen was also lost. NAD plus comes in and takes that hydrogen and converts NAD plus to NADH. This molecule of NADH is then shipped off to the electron transport chain. Okay, we are here with a 4-carbon structure. 4-carbon structure, we'll look at the other side. This is also a 4-carbon structure. So there were no carbons lost this time. 4-carbon went to 4-carbon. The only thing that happened here was molecular rearrangement. Some of those molecules moved around. During this process, ADP, one molecule of ADP, was put into the system along with PI, which is inorganic phosphate, and was converted to ATP. This molecule of ATP is energy in its final form. It does not have to be shipped off to the electron transport chain. It is ready to be used. Okay, next we have a 4-carbon backbone. And over here, the next product is a 4-carbon backbone. Again, no carbons were lost. The only thing that happened was molecular rearrangement. But now notice, we see for the first and only time, we see a new electron acceptor. That electron acceptor is FAD. This was mentioned in my last lecture because it is an electron carrier. It carries electrons in the form of hydrogen. But if you notice, it picks up 2 hydrogens, okay? So this has technically, the technical term is it's been reduced because it gained electrons. The FAD has been reduced to FADH2. It has picked up those 2 hydrogens, and each hydrogen is an electron, so it picked up 2 electrons. All right, this molecule of FADH2, once it's made, it is then shipped off to the electron transport chain. Okay, we are here with the 4-carbon backbone structure, going up to the next 4-carbon backbone structure, and we see that during this process more hydrogen was lost. The NAD plus was reduced to NADH because it gained a hydrogen. It gained an electron in the form of that hydrogen. This molecule of NADH was then shipped off to the electron transport chain. Okay, so we are here now. We are with OAA. We are back to our starting point, oxaloacetyl acetate. It will take the next incoming acetyl group to form citric acid, and the entire pathway will start over again. Okay. So that is the Krebs cycle. So let's talk about what we've made here. Looking at just this one slide, we have made 1 molecule of NADH, 2, 3, we've made 4 molecules of NADH. And you bear with me, I'm writing using a laptop and it's challenging. So we made 4 molecules of NADH. Okay, we made 1 molecule of ATP. And we made 1 molecule of FADH2. Okay, I'm about to tell you something that you need to write down. Write it in your notes as you are listening to this. Each molecule of NADH will yield 3 molecules of ATP in the electron transport chain. So again, I'll repeat that. Each molecule of NADH will yield 3 molecules of ATP in the electron transport chain. Okay. Now, the second fact that you need to write down. Each molecule of FADH2 yields 2 molecules of ATP in the electron transport chain. So again, each molecule of each molecule of FADH2 will yield 2 molecules of ATP in the electron transport chain. Okay. So if we were to go through here and if we were to count our energy, we have an NADH here at the top, and I'm going to change ink colors because this is looking a little sloppy. Okay. So this molecule of NADH, remember, it makes 3 molecules of ATP. So that's 3. 6, 9, 12. Okay. Now, FADH2 only makes 2 molecules of ATP. So 12 plus 2 is 14. Okay. So far we're at 14. And then at the very bottom down here with ATP, we have 1. 15. So, again, each molecule of NADH yields 3 molecules of ATP, and each molecule of FADH2 yields 2 molecules of ATP. So if you count your NADHs, each NADH is 3 ATP. Your molecule of FADH2 is 2 ATP. And then you have 1 lone molecule of plain regular ATP at the bottom. So on this picture right here, this picture is showing a total of 15 molecules of ATP. However, remember from the Krebs cycle, remember from glycolysis, that molecule, fructose 1,6-biphosphate, it got cut in half. And I said that each half of the molecule became glyceraldehyde 3-phosphate. And so I said then to take all your numbers and multiply them by 2. Okay? So if we have 15, we have to take 15 and we multiply it by 2. Okay? So we get 30. So we get 30 molecules of ATP from the Krebs cycle, from this slide right here. Okay? And then if you were to go back and count the energy that was made during glycolysis, well, you make 10 molecules of ATP in glycolysis. And you can easily figure this out by going back and counting the NADH that was made and the ATP that was made. So you make 10 molecules of ATP during glycolysis. You make 30 molecules of ATP during the Krebs cycle. So if you add 10 and 30 together, you get 40. However, remember, you had to use 2 molecules of ATP to get the entire process started. So 40 minus 2 is 38. So this is a test question. Per molecule of glucose, how much ATP is made, or what is the net ATP that is made? The answer is 38 molecules of ATP per molecule of glucose. 38 ATP per molecule of glucose. Now, in A&P, you may remember that number being 36, and that is true. But the difference is the cell type. In eukaryotes, a molecule of glucose only yields 36 molecules of ATP. But because we are dealing with prokaryotes, such as the bacteria, each molecule of glucose yields 38 molecules of ATP. Okay. All right. As always, if you don't understand or you have questions, feel free to message me and ask for help. Okay, the third and final process is the electron transport chain. It's called the electron transport chain because it is literally a chain. Okay? It is formed by something known as cytochromes. You will see this on the next slide. Cytochromes, these are for the most part, cytochromes are proteins, and this is what forms the electron transport chain. Make sure you notice the location of the electron transport chain. The location of the electron transport chain is in the plasma membrane. Now, if you compare that to in a eukaryotic cell, the electron transport chain is located in the mitochondria. But remember, prokaryotes, they don't have mitochondria. So the electron transport chain is located in that plasma membrane, that phospholipid bilayer that we talked about. And I don't want you to think that there's just one electron transport chain per cell, because that is not true. There's 50 to thousands of electron transport chains that are located in the plasma membrane of the bacteria cell. So this is where the oxidation reduction reactions happen. You will see a lot of phosphorylation here. The electron transport chain also generates what's known as a proton motive force, or a proton gradient. Because remember, okay, let me write something real quick. If we have hydrogen, you are going to see on the next slide, you are going to see a lot of H pluses. Okay, remember, hydrogen has one proton, it has one electron. Okay, we've been thinking of hydrogens as being electron carriers, but now they're going to have a slightly different function. You're going to see H pluses. The only way a hydrogen can have a positive charge is if it loses its electron. And you are going to see a gradient. And what that means is you are going to see a lot of hydrogen, a lot of plus charged hydrogen ions on the other side of the plasma membrane. These are thought of as protons now because it is a hydrogen that has lost its electron. So literally all that's left is a hydrogen and its proton, which is a positive charge. So now whenever you see these hydrogen ions, these positively charged hydrogen ions, you have to think of them as protons. And so you are going to see a proton gradient on the next slide. And then you will, like I said, you will see oxidative phosphorylation. So let's move ahead and let's put all this in picture form. Simply, I really like pictures. I think it's easier to explain using pictures. Okay, first, let's look at this picture in the upper right-hand corner. Okay. This is a simplified version of the electron transport chain. I pulled this picture off of Google because I was looking for something very simple to introduce you to the electron transport chain with. Okay, so this is the electron transport chain. It is located in the plasma membrane. I said most of it is composed of something known as cytochromes. So if you look at the naming of this, it's named FMN, Q, cytochrome B, cytochrome C sub 1, cytochrome C, cytochrome A sub 3. You do not have to know the names of the cytochromes and the order in which they are in. But this just goes to point out what I said on the previous slide, that most of the components of the electron transport chain are protein-based. These cytochromes are proteins. Okay. Now the first one up here, FMN is not a protein. It's actually a metal. But all the others, Q and all of the cytochromes, these are all proteins. Now look at something at the very end. You see oxygen. That oxygen has a very important name. At the end of every electron transport chain, you have what is known as a terminal electron acceptor. For aerobic respiration, the terminal electron acceptor is always oxygen. So what that means, at the end of every electron transport chain this is for aerobic bacteria only. I'll stop there and recap. Remember, there's 3 types of respiration. You have aerobic respiration, you have anaerobic respiration, and then you have fermentation. I said 90% of my talk would be on aerobic respiration. So these are the bacteria that need oxygen to survive. So this picture here is talking about the situation or the setup for these aerobic bacteria. At the very end of the electron transport chain, you will have oxygen. Oxygen is the terminal electron acceptor for aerobic respiration. Okay. You will also, by the end of this chapter, you will also be introduced to the terminal electron acceptor for anaerobic respiration and for fermentation. And you need to know all those because they are test questions. So I will repeat myself one last time before I move on. Oxygen is the terminal electron acceptor for aerobic respiration. Oxygen at the very end, you see there's hydrogens being put into the electron transport chain. Well, those oxygens are going to bind to the incoming hydrogen and they're going to form water. So water is always considered to be a waste product of the electron transport chain. Okay, now that is the simplified version of the electron transport chain. Now let's look at the more complicated version of the electron transport chain. And although it's more complicated, there's a lot more stuff going on, I really, really like this picture. They did a very good job making this picture. And before I continue, I will let you know this picture down here at the bottom, this entire picture will be on your next exam. And it will be there's like 3 or 4 multiple choice questions that I ask you based on that picture. Okay, so let's go ahead and get started. Okay, so first of all, note on this picture here that the electron transport chain is located in the plasma membrane because you see the phospholipid bilayer. Okay, it goes in the same order: FMN, Q, cytochrome B, cytochrome C sub 1, as the other one up here. It's arranged a little bit differently. All right, another thing I want to point out, there's an enzyme here at the very end in blue. The name of that enzyme is called ATP synthase, and the spelling of it is down here. Make sure you know the name of the enzyme and that you know what it does. All right, and we'll talk about what it does in just a minute. So if you notice here, there is a number 1. That means we are going to start right here. All right. During glycolysis and during the Krebs cycle, we made a whole bunch of molecules of NADH and FADH2. And I said, every time one was made, I said it was shipped off to the electron transport chain. Well, that is what step 1 is representing. This molecule of NADH was one that was made in either glycolysis or the Krebs cycle, and then this was shipped off to the electron transport chain. So this is step number 1. It is the starting point of the electron transport chain. And there's a lot going on here. We have, once again, we have 3 different things going on here. So I'm going to start by talking about this arrow here. Okay, you notice that NADH lost that hydrogen. That means it lost the electron. Okay, so it was oxidized. NADH is oxidized back to NAD+. The only thing that happened here is that hydrogen right there was lost. NADH is converted back, or you could say the technical term, oxidized, back to NAD+. This molecule of NAD+ is now ready to be used again. It will travel back up to glycolysis or back up to the Krebs cycle and take on more hydrogens, and it'll be reused over and over again. But now let's follow this other black line, just the black line. Okay, so it lost this hydrogen right here. That last hydrogen was lost. Well, you have to ask yourself, if that hydrogen was lost, where did the hydrogen go? Okay, here's the answer is this. That hydrogen went through FMN to the other side of the plasma membrane, because you see all these hydrogens are being pushed to the other side. Okay. So the hydrogen that was lost off of NADH, that hydrogen was pushed through FMN, because there are channel pores that go through FMN, and that hydrogen is pushed through FMN to the other side of the plasma membrane. Okay, now, so you have a whole bunch of hydrogens over on this side, but not that many hydrogens on the other side because they've all been pushed over to the other side. Okay, now, notice all these hydrogens are gathered over here. But look at those actual hydrogens. It says H+. That is a proton. Remember from the previous slide, I said that an H+ is a proton. You have a proton gradient now, because that is exactly what a gradient is. It's where you have a lot of something on one side and not much of that same something on the other side. So on this side of the plasma membrane, you have a whole bunch of hydrogens, but you don't have that many hydrogens on this side of the plasma membrane, because all the hydrogens, once again, have been pushed through FMN to the other side. Okay, the next thing you have to ask yourself, well, those hydrogens, they carry a positive charge. That means it lost its electron. Well, if it lost its electron, you have to ask yourself, where did the electron go? Well, now follow the red line. So if I turn on my highlighter and I follow the red line, the red line is where the electron goes off of that hydrogen. That is the only way that a hydrogen can have a plus charge, is if it lost its electron. And it did lose its electron. That is what that red line is showing. It's showing you where that electron is going. That is why this is called an electron transport chain. It transports those electrons lost by those hydrogens. Okay, here you see oxygen. That is this oxygen right here. That is what's known as the terminal electron acceptor. Okay. It's going to bind to incoming molecules of hydrogen, and it's going to form water as a waste product. Okay, so let's go back to our hydrogen gradient. There's a bunch of hydrogens over here. On this side, we have a whole bunch of hydrogens. On this side of the plasma membrane, and we have very few hydrogens on the other side of the plasma membrane. Well, hydrogens, they want to be at equilibrium with one another. What that means is there needs to be, or the hydrogens, they want an equal concentration of hydrogens on both sides of the plasma membrane. However, there are a bunch of hydrogens here, and this is the periplasmic space. Okay. So there's a bunch of hydrogens on this side, not a lot of hydrogens on this side, on the bottom part. Hydrogens can come over to the other side of the plasma membrane, but in order to do that, you see right here, hydrogens want to go from one side of the plasma membrane to the other, and they can, but only by traveling through this enzyme known as ATP synthase. Okay, what did I tell you about enzymes? First of all, they usually end in ASE, and they usually tell you what they do. ATP synthase, well, that sounds like you're synthesizing ATP. And that's correct. That's exactly what you're doing. You are synthesizing ATP. So, 3 hydrogens enter here at the top of ATP synthase. 1 enters at a time, and then this top part is like a rotor. This top part moves. So 1 hydrogen enters, the top part of the enzyme moves. A second hydrogen will enter, the top part of the enzyme moves again. The third hydrogen enters, the top part moves again. When 3 hydrogens have entered, there is now enough energy in that molecule, in that enzyme molecule, for the bottom part of the enzyme to turn once, 1 complete turn. Now, ADP molecules, see these ADP right here. Those ADP that I just highlighted in purple, they are stuck to the bottom part of the rotor. When the bottom turns 1 complete turn, it allows free inorganic phosphate to bind to the ADP that's stuck on the bottom of the rotor to form ATP. And that is how the electron transport chain works to make energy, to make ATP. Remember, the goal of this chapter was to make ATP. And this is what we've done. This is the final step of everything that I've talked about. And you can see that we have made ATP in yellow. So each molecule of NADH yields 3 molecules of ATP. Now, along with this video, I am sending a YouTube link to a video that I show my students every semester. It's talking about the enzyme ATP synthase, and it animates it for you. So you can see how 3 molecules of hydrogen enter into the top part of ATP synthase, and how the rotor turns, and how ADP is converted into ATP. It's a really nice video. It's only 2 or 3 minutes long. Make sure to watch the video, and you will have a better understanding of how the electron transport chain works and how this enzyme ATP synthase works. So on the exam, I told you I would have this exact picture of the exam on the exam, and I would ask you a couple of things. I would ask you, okay, where is water made? And again, water is made here. Okay. I ask you the location of the electron transport chain, and remember, it's located in the plasma membrane. I ask you the name of the enzyme ATP synthase and what it does. Well, it makes ATP from ADP. And then I ask you What is the terminal electron acceptor for aerobic respiration? And it's this, it's oxygen. Okay. So there's a lot going on this slide, but you know I'm always available through email or through the Remind app if you have any questions. Okay, so that is the end of aerobic respiration. We have covered all 26 to 28 steps of aerobic respiration. We have started off with glycolysis and we started off with glucose. And then we have went through all the steps and we wound up with making ATP, which is energy. Okay, so if we move on to the next slide. Everything on the left side here under respiration, we've already covered everything on this. This is just a review. You start off with glycolysis. Okay. Glucose enters into glycolysis. You form pyruvic acid. During glycolysis, you make a little bit of NADH. You make a little bit of ATP. The pyruvic acid is converted into acetyl-CoA. The acetyl group enters into the Krebs cycle, and during the Krebs cycle, this is where CO2 is made, ATP is made, NADH is made, and also it's missing something. Remember, we made one molecule of FADH2 here, and it's not being shown, but we did make one molecule of FADH2. And then all those molecules of NADH and FADH2 are then shipped off to the electron transport chain. The electron transport chain is where the NADHs lose their hydrogen. They're shipped to the other side of the plasma membrane and they wind up making or adding on a phosphate to the ADP that's stuck on ATP synthase, making ATP, which is seen here in yellow. You see here oxygen is the terminal electron acceptor and you have water as a waste product. So this is a complete review of everything that I've talked about. What we have not talked about yet is fermentation. And I have very little to say on fermentation because I said 90% of my talk would be on aerobic respiration. So some bacteria have the ability to undergo respiration and fermentation. For example, E. coli. E. coli can undergo respiration, but it can also undergo fermentation. And I also said early on that if you understood aerobic respiration, you would automatically understand anaerobic respiration and fermentation. And the reason is, well, you see with fermentation, it goes through the same beginning as respiration does, and then it just stops. So with fermentation, it still starts off with glucose. Glucose goes through glycolysis. You wind up with pyruvic acid, just like we saw with respiration. But with fermentation, the pyruvic acid undergoes fermentation to produce end products. Now, there's a list of about 30 different end products that are made. However, there's really only 2 end products that I'm interested in you knowing. One is ethanol. This is drinking alcohol. Okay? So ethanol is one common end product of fermentation. Another common end product of fermentation is lactic acid. And remember, this is the bacteria that's added to your yogurt because it produces lactic acid, making your yogurt have that tart, tangy taste. So lactic acid is really important in the food industry. So again, this is the process of fermentation. It's very simple. Fermentation undergoes glycolysis, and then it stops. It does not go through the Krebs cycle at all. Okay. So again, fermentation goes through glycolysis, and it stops. It does not undergo the Krebs cycle at all. So fermentation makes very little energy. The only energy that comes from fermentation is the energy made during glycolysis. And remember, there wasn't much made in glycolysis. We made a little bit, very little bit of NADH, and we made a couple of molecules of ATP, and that's it. So fermentation, yes, it is technically a way of making energy, but it's a very poor way of making energy. And I have one more slide where we'll talk about fermentation. later on in a few minutes. Okay, moving on to the next slide. Okay, this is classically one of your 5-point questions on your exam. This exact picture will be on your exam, and this is your 5-point question. I give you this entire column. I give you this information where I'm making the purple mark above it. I give you all of that information. But here in yellow, what I'm about to highlight in yellow, I leave that part blank. I leave the boxes there, but I mark out the lettering. So you have to know in each box, you have to know what goes in each box. And so this is basically just telling you where that energy comes from. And over here in the last column, for example, it says 6 ATP through electron transport and chemiosmosis. For your exam, all you have to tell me is 6 ATP. You do not have to tell me through electron transport and chemiosmosis. For any of those, you do not have to tell me through electron transport and chemiosmosis. You have to give me a number, and you have to give me what kind of energy it is, whether it's NADH, whether it's FADH2, or whether it's ATP. So this is to summarize how I was telling you where all that energy comes from. So during glycolysis, remember we made 2 molecules of NADH. And then in pyruvic acid, during that transition step of pyruvic acid being converted into acetyl-CoA, we made two more molecules of NADH. And then in the Krebs cycle, we made 3 molecules of NADH, and we made 2 molecules of FADH2. Well, if you go back to glycolysis, you will see that we made 2 molecules of ATP directly. Okay? And then if you remember my rule, each molecule of NADH makes 3 molecules of ATP. So 2 times 3 is 6. 2 times 3 is 6. And then you get down to the Krebs cycle, and in the Krebs cycle you make 2 molecules of ATP directly. And then you make 6 molecules of NADH. And remember, 6 times 3 is 18. My other rule that I had you write down was for each molecule of FADH2, it yielded 2 molecules of ATP. So 2 times 2 is 4. So if you add all those numbers together, you get 38 molecules of ATP per molecule of glucose. Okay, what we have not talked about yet, we have not talked about anaerobic respiration. Remember, I was saying that most bacteria are a lot like us. Most bacteria like oxygen. They need oxygen to grow. They do very well with oxygen present. However, there are some bacteria that die in the presence of oxygen. Oxygen is toxic for certain bacteria. Okay, that is what anaerobic respiration is. This is how these anaerobic bacteria make their energy or make their ATP. Well, the first thing you need to look at is that final electron acceptor. In the previous slide, I called it a terminal electron acceptor. It's the same thing. Final electron acceptor, terminal electron acceptor, again, it's the same thing either way. When we were talking about aerobic respiration, remember that the final electron acceptor for aerobic respiration is oxygen. Well, we can't use oxygen as a final electron acceptor in anaerobic respiration because oxygen kills the bacteria. So instead of using oxygen, we have to use something else. So the terminal electron acceptor, or that final electron acceptor for anaerobic respiration, is either some type of nitrate or some type of sulfate. Okay. So this is your second category of terminal electron acceptors that we've talked about. So again, to recap at this point, your final electron acceptor for aerobic respiration is oxygen, always oxygen. Your final electron acceptor for anaerobic respiration is usually something nitrogen-based or sulfur-based. Okay, as far as how much energy is made, well, if you look down here at the bottom, ATP yield is lower than that in aerobic respiration because only part of the Krebs cycle operates under anaerobic conditions. Okay, what that means is only part of the Krebs cycle will function if there's no oxygen present. Okay. So you're not going to make as much energy with anaerobic respiration as you do with aerobic respiration. So we've talked about the three. We've talked about aerobic respiration, anaerobic respiration, and fermentation now. So if we were to categorize them, or if we were going to list them in order of which produce the most ATP, well, aerobic respiration produces the most ATP, followed by anaerobic respiration, followed by fermentation. So fermentation produces the least amount of ATP. Okay, so again, with anaerobic respiration, the key here, glycolysis proceeds as normal. You don't need oxygen in glycolysis. Glycolysis proceeds as normal. But only part of the Krebs cycle can function where there's no oxygen. So only part of the Krebs cycle works. And then the terminal electron acceptor cannot be oxygen with anaerobic respiration. So if it can't be oxygen, well, there still has to be a terminal electron acceptor or a final electron acceptor, but it uses something besides plain oxygen. It uses some type of nitrate or some type of sulfate. Okay, this is my only other slide on fermentation. I briefly talked about fermentation a few minutes ago. So fermentation gives you a little bit of general knowledge of fermentation is at the top of the slide. It's fermentation, any spoilage of food by microorganism. We use it a lot to produce alcoholic beverages or acidic dairy products. We're able to use it on a large scale basis, particularly in industry. But what I'm really interested in is knowing the scientific definition of fermentation. Okay, the first one is key. It uses an organic molecule as the final electron acceptor. So now we have officially covered all three categories of final electron acceptors. For aerobic respiration, it's oxygen. For anaerobic respiration, it's nitrogen or sulfate. For fermentation, it's any organic molecule. And remember, organic just means that it contains carbon. Okay? Fermentation does not use the Krebs cycle or the electron transport chain, and therefore the energy yield is super low. Really, the only energy that's going to be made is just a tiny bit in glycolysis. And there is a diversity of end products. Usually there's about 30 different end products that can be made, but I'm only interested in you knowing the two I have listed, lactic acid and ethanol. Okay, let's talk about catabolism, which is the breakdown of other compounds. So this is a list of some polysaccharides and disaccharides. Let's go down to the very bottom and talk about the disaccharides first, simply because we've already talked about the disaccharides a little bit. Remember at the start of this chapter we talked about sucrose? Well, sucrose is a disaccharide because if you break that bond that holds those two smaller molecules to form sucrose, it breaks down into glucose and fructose. So sucrose is a disaccharide. We've already talked about those. We've already talked about how you make energy from those. Going back up to talk about the amylase. Okay, amylase is an enzyme. Amylases, this is a plural word. The singular word is amylase. It ends in ASE, so you know it's an enzyme. This is used for the digestion of starch, which is very, very common. Remember, starch is nothing more than large, massive chains of glucose. We use glucose, but the starch molecule is way too big. We can't use starch for anything. So the function of these amylases is to break down these large molecules of starch into individual molecules of glucose. And once we have individual molecules of glucose, it will go straight into glycolysis and make energies just like we talked about. So one of the main places that you find amylase in humans is in our saliva, because as we eat food, as we eat starches, they meet with these amylases and they start being broken down. Okay, so as far as the those enzymes we were talking about, cellulase breaks down cellulose. However, most humans, as well as most animals, we don't have the enzyme cellulase. We still consume cellulose, but it's usually the bacteria in our gut that actually breaks down the actual cellulose. So although cellulase is a very common enzyme, it's a very necessary enzyme, we simply don't have that enzyme. Moving on to protein catabolism. Okay, so not everything can be broken down into sugar. Yes, sugar is great. Your starches, your carbohydrates can be broken down into sugars. But if you eat a piece of meat, if you eat a steak, well, that steak is high in protein, and there's no way you can convert a protein into sugar. But we still need energy from protein. So if you look at what happens with protein, first of all, that protein is broken down. I've said before that protein is nothing more than a chain of amino acids. You take that protein, it's exposed to something known as proteases. So proteases, these are enzymes that break down proteins. So this protein is broken down into extracellular proteases, and you wind up, what's left is with nothing more than an amino acid. And then that amino acid, okay, so here is the basic structure of an amino acid. Remember, there are 20 amino acids. You have a central carbon. This is known as a carboxyl group. You have your amino group. Your R is your side chain. Okay, so every amino acid has the same structure. The only thing that's different between the 20 different amino acids is that R chain. That R takes on one of 20 different forms, and it will give you a different amino acid. So once you have that amino acid, it can undergo a wide variety of things as seen up here. Deamination means this bond is broken right here. So maybe if I switch color, okay. Deamination is the breaking of this bond. Decarboxylation is the breaking of this bond. Dehydrogenation is the breakage of any hydrogen. So you could break that one, or you could break that one. Desulfurylation, this is the removal or the breaking of sulfur. Now, by looking at that basic amino acid structure, you don't see sulfur in there. But if you look at the amino acids, if you look at the R group of all 20 amino acids, we actually have a handful. There's 4 or 5 different amino acids that have sulfur in there in the R group. So that is what that desulfurylation is talking about, the breakage of sulfur bonds in that R group. So you take this amino acid and you break it down. What you're left with is simply called an organic acid. Nothing fancy. It's just an organic acid. And then these organic acids enter straight into the Krebs cycle. So that is how you get energy from protein. Protein does not go through glycolysis at all. The protein's broken down to amino acids, which is then broken down into smaller organic acids, which then enter straight into the Krebs cycle. So yes, you still get energy from protein, okay? Just not as much energy, because you get more energy from sugars because glycolysis is there, and you do make a little bit of sugar or a little bit of energy in glycolysis. So that is how you'd go about breaking down proteins. Okay, fermentation. This is the test, or this is the virtual lab that I had you guys doing this week. Fermentation, The purpose of a fermentation test is to see if a bacteria can ferment a particular sugar. Okay, so this picture here is looking for mannitol fermentation. So in tube A, down here at the very bottom, tube A, the red media, it's called phenol red, and there's a pH indicator built in to the phenol red media. There is a little tube on the inside that is known as a Durham tube. D-U-R-H-A-M. Durham tube. So that little glass tube on the inside, and you notice it's a small glass tube and it's upside down. Okay. The reason it's upside down is it's going to collect any gas if gas is produced during the fermentation process. So again, you have a test tube with phenol red media, and there's a pH indicator built into the phenol red, and then you have a Durham tube that's upside down in your test tube. Well, this test is testing for mannitol. So what that means is mannitol is a sugar. That means the only sugar source that is in test tube A is mannitol. That's the only sugar available. So this test is testing to see if your bacteria can ferment mannitol, meaning can your bacteria use mannitol as a carbon source? And so I'm going to take a bacteria, random bacteria. I'm just going to use the example E. coli. I'm going to inoculate E. coli in test tube A. Okay, so E. coli has been inoculated in test tube A, and then I incubate test tube A in an incubator for 24 to 48 hours. These are my possible results. I may get a result that looks like B. I may get a result that looks like C. I may get a result that looks like D. What B means, if you go back and you get your test tube out of the incubator, well, there's not really any color change. It's still kind of a red. It's still red for the most part. There's no gas bubble in the Durham tube. So that means fermentation did not occur. If you move over to C, test tube C, if this was your result, was kind of an orange color. And if you look at your Durham tube, there is a tiny, tiny gas bubble in there. But according to the way this test works, orange is also considered to be a negative test. So this is a negative test here. Okay. Now, if I went back and got my test tube out and it looked like D, so it went from A to D, well, that is what's considered lemon yellow, bright lemon yellow. And if you look at the Durham tube, there's a huge gas bubble in the top. So it changed colors and it produced gas. So if that were my result, then I could say it's positive for mannitol fermentation, meaning my bacteria, and remember I just used E. coli as an example, my E. coli was successfully able to ferment mannitol, and I know this because it produced acid. Acid caused the color change because there's a pH indicator in the phenol red media, and it produced gas, and the usual gas is carbon dioxide. And you can see the carbon dioxide bubble. There's a big gas bubble in that Durham tube. So D is a positive result. So B is negative, C is negative, D is positive. And that's the way the fermentation tests work. Okay, last slide in this chapter, metabolic diversity among organisms. Well, you have some new terms. You have phototroph, you have chemotroph, you have autotroph, you have heterotroph. However, the only one that I'm really interested in you knowing is the word in red, chemoheterotroph. And I said you've already learned it. You will see it multiple times between now and the end of the semester. A chemoheterotroph is any organism that uses the same organic compound as an energy source and a carbon source. It's the most medically important bacteria. Okay, humans, we are chemoheterotrophs. Pretty much all your animals are chemoheterotrophs. What is not a chemoheterotroph? Any organism that undergoes photosynthesis, such as your plants, your algae, they are not chemoheterotrophs. Okay, so a chemoheterotroph is any organism that eats carbon and gets energy from the carbon that they eat. So we eat carbon all the time, every day. Most of our foods are carbon. Most of the foods that bacteria eat are carbon, and they're able to use this to get energy from. Therefore, that is the definition of chemoheterotroph. And that is the end of chapter 8. I will send you chapter 9 tomorrow.